That's awesome. All dues. Hello. Oh, you can.
Okay, so short times we have not put it on.
Yeah.
I'm just gonna do. I know, right?
Thanks. About that, I forget again.
I just found her.
Either way.
Yeah, for sure.
Yeah, it's wide open.
They'll drop it on the ground.
You should have your same kind of belt. Yeah, absolutely. Okay, yeah. Uh-huh.
The other one had she's side by side.
Well, good afternoon, everyone. I think we'll get started. A few people may file in the coming minutes, but most of you know me. I'm David Ehrman, Neuromuscular Division Chief here at University of Rochester, and it's really my honor to do the introduction for a very special Grand Rounds. This is the 9th annual Dr. Richard T. Moxley III Rare Disease Lectureship. And while many of you in this room and online know Dr. Moxley, I realize there are many of you who haven't met Dr. Moxley or may not be aware of his contributions to our department and to neuromuscular writ large. Dr. Moxley, we're very fortunate to have you online today. I hope you can hear me and hear all of us. So by way of a bit of background to Dr. Moxley, and we know Dr. Moxley more affectionately as Mox, so I'll use the name Mox. But Mox did his formative training at Harvard and at Penn, and after a research fellowship at the NIH, we were fortunate to have him join our department in 1974. And he was on faculty for 43 years, retiring in 2017, and continues as emeritus faculty. And his contributions to our field, to the University of Rochester Medical Center, to the department are innumerable, too numerous to really mention today. But just a few very specific highlights. Mox contributed immeasurably in the field of rare diseases, and hence the Rare Diseases Lectureship that bears his name. In the 1980s, he collaborated with Sri Pandya and Birch Griggs to develop and demonstrate that corticosteroids are effective for Duchenne muscular dystrophy therapy. This resulted in a seminal publication in 1989 in the New England Journal of Medicine, and corticosteroids are a mainstay of Duchenne muscular dystrophy therapy today. He also devoted his career to understanding the myotonic dystrophies. He was instrumental collaborating with Charles Thornton in really deep phenotyping myotonic dystrophy, describing myotonic dystrophy type 2 or proximal myotonic myopathy, and then working closely with Charles to elucidate the disease pathomechanisms. And then Mox really set up the initial infrastructure to conduct clinical trials in myotonic dystrophy. He had the foresight to get successfully funded through and obtain one of the initial Senator Paul Wellstone Muscular Dystrophy Research Center grants. And we continue to have that funding today through the efforts of Charles Thornton and Johanna Hummel and others. And so that led to everything we have seen today in terms of us being on the brink of clinical therapies for myotonic dystrophy. But beyond his research contributions, he contributed at a leadership level in our department, being the associate chair from 1997 to 2012. He was a leader in clinical research at the institution and was director of our clinical research center for a number of years. And perhaps his greatest impact has been the impact on all of us. He was a mentor to myself and a mentor to many others in the department, both past and present. And so in his name, we developed this lectureship in 2017. He's contributed to the sustainability of the lectureship. And we've had a wonderful set of lecturers starting in 2018 with Jerry Mendel, who we all remember speaking to us at the inception of gene therapy for SMA and Duchenne muscular dystrophy. And since that year, we've seen a multitude of invited lecturers who've been leaders in the field. And so without further ado, I'd like to turn it over to Mox for any introductory remarks, and then we'll have Peter Cree introduce our 9th annual Dr. Moxley invited lecturer.
Well, David, I really am without words, and I know a lot of you find that hard to believe, but thank you so much for that generous introduction and for all the work that you and your colleagues have done to make this lectureship possible. And I look very much forward to what we're going to hear from Dr. Shneider today, and insights into not just ALS, but perhaps other anterior horn cell disorders. And I just feel very good about the overall impact of what Joan and I are trying to do with donations to help research move forward. So I've said more than I needed to. Just thank you to all the people who've helped me connect with the video and the Zoom. And I look forward to the lecture, and I'm just going to mute myself again.
All right. Thank you, Mox. We look forward to your questions at the end. Mox always has the most insightful questions. So without further ado, I'll turn it over to Dr. Peter Cree, who, as you know, is director of our ALS program, to introduce Dr. Shneider.
Okay. Thank you, David. Thank you, Mox. So it is my privilege to introduce this year's lecturer, Dr. Neil Shneider. Dr. Shneider completed his undergrad training at Harvard College and earned both his MD and PhD at Columbia University. He trained with some of the most distinguished leaders in neuroscience, including Nobel laureate Richard Axel and Thomas Jessel, and later served as staff scientist at the NIH. He is now the Claire Tao Professor of Motor Neuron Disorders at Columbia University and the director of the Eleanor and Lou Gehrig's ALS Center. In that role, he leads a remarkable program that spans the full spectrum of ALS research, from fundamental disease mechanisms to translational models to clinical investigation and therapeutic discovery. His work has focused on major genetic drivers of ALS, including FUS and C9orf72, and helped push the field towards a deeper understanding of ALS. Over the last few years, I've had the pleasure of getting to know Dr. Shneider through our work together on a statewide initiative to make New York a national leader in access to the highest level of ALS care and research opportunities. I have already learned a great deal from him, and I look very much forward to continuing to build that work together. Through that collaboration, I have developed a deep appreciation not only for the innovation, rigor, and integrity that defines his scientific and clinical work, but also for his genuine empathy towards patients and colleagues, and of course, for his pointed sense of humor. ALS is a rare disease, but it is a field with many strong voices and many competing directions. That diversity is important, but we are especially fortunate that Dr. Shneider has not simply followed the field's established currents. Instead, he has often chosen his own course, at times upwind, in a direction that has already made a profound impact, and I believe will continue to have an even greater one for patients and families affected by this devastating disease. We are grateful for his work, honored to have him with us today, and very much look forward to hearing him share some of that work with us. Dr. Shneider, thank you and welcome.
You want to help me? Yeah. Well, I'll begin by thanking Peter for that really generous introduction. Thank you, Dr. Holloway, Dr. Herman, Jackie, you're here. Thank you for shepherding me through this and for this really wonderful invitation to honor Dr. Moxley personally and honor his legacy to the field. Here. So, you know, when I began reading about Dr. Moxley, I really never had the pleasure of meeting him until this morning. I learned that as he was telling the crowd, I'm not sure he knew he was on mic earlier, but that he and I have a lot in common, not just a haircut, but we're both graduates, as you heard, of Harvard College. We both trained really in the wonderful Harvard Longwood Neurology training program, both had the same really extraordinary experience there. And, you know, when I was thinking and looking over his career, is that he and I too trained at a moment when neurology was at a very different place. It was a field that was largely, you know, observational. We focused on phenomenology. We focused on characterization. We focused on diagnosis and management. And it was hard for me, and I'm sure for him and his generation, to imagine the moment that we're in, you know, today, where neurology really is in a very different place, a more interventional discipline, where we can offer therapeutics that are molecular, in many cases gene-based, and based on a growing understanding of the biology of the disease. So these are rational therapeutics that we're bringing into the clinic that have the potential to really change the natural history of these diseases. And again, as a resident and through much of my career, I never really, you know, thought that we would be at this place, to be honest, and certainly didn't expect to play the role that I have, and I'm happy to share my experience with you today. So, like many of you here at this wonderful Center for Neuromuscular Research and Neurological Research, my work begins and ends with patients. We study our patients, we sequence them, we study their genomics, we collect samples from them. We use the genetics to create animal and cellular models of disease, And we study those models to discover mechanisms and pathways of interest, identify targets, and then develop therapeutics, some gene-based, small molecules, a variety of modalities that we're now bringing to bear on neurological diseases. And we bring these therapeutics back into the clinic for the benefits of our patients and their families. And so this has generally been very much true of ALS, which I'm sorry to say still in 2026 is still a deadly neurological disease in most cases. Fatal neurodegenerative disorder, predominantly involving motor neurons, but of course it's not only a motor neuron disease. It's a multi-system, multicellular disorder that has very close associations, genetic, pathological, clinical associations with frontotemporal dementia. In the United States, the prevalence of this disease is about 9 to 10 per 100,000, reflecting an incidence of about 1 to 2 per 100,000. Small prevalence given this incidence because of the short survival. Most of our patients don't survive 3 years, and only a small fraction, less than 10%, beyond 10 years. Most of our patients have sporadic disease, meaning they have no known family history. In the remaining 10 to 15% are associated with an ALS gene mutation. Most of those are familial, but we have many cases of sporadic disease in which we find gene mutations. Over the course of the last 30 years, we've discovered more than 30 ALS FTD-associated genes represented here. These account for nearly 80% are now of familial cases. There are still some left to be identified. And as I said, many of these same variants and these same genes are identified in the sporadic patients, either because of unknown family history or incomplete family history, or because of de novo spontaneous mutations in these same genes, which we find repeatedly, as you'll see. So this is a figure I like, many versions of this, but it sort of shows the history of ALS gene discovery, beginning in 1993 with the discovery of SOD1. Bob Brown, Tipu Siddiq, and their colleagues identified variants in this gene, superoxide dismutase 1, that are responsible for familial ALS. And over the course of the years, the pace of discovery increasing with the publication of the human genome and developments, next-gen sequencing technologies, the pace of discovery increased significantly in the early 2000s and 2010s, include things like C9orf72, the most common genetic form of ALS and frontotemporal dementia. 1993 was the discovery of the SOD1 mutations, and almost exactly 30 years to that date, Qalsody or tofersen was approved by the FDA. This was an absolute milestone, I think, in the history of ALS. The first ALS therapeutic, the first gene-based therapeutic, or really ALS therapeutic of any kind, to really alter the natural history of this disease. And that was true not only in terms of its lowering of neurofilament light, which is a marker of neuronal injury, not specific to, but very useful, as you'll hear in ALS, but also in terms of slowing the rate of progression. And we know now from post-marketing studies that not only does it slow the rate of progression, but in upwards of 20% or so of treated patients, we're seeing some degree of functional recovery, absolutely extraordinary. And this is manifesting in real effects on survival. It is the first example of this, and I think, as I'll try to convince you today, other cases will follow. So many ALS genes that are potential targets for this kind of therapeutic intervention. I'll talk to you today about my efforts on one of them, FUS, fused in sarcoma. This is a gene in which ALS-associated variants were first identified in 2009 by Bob Brown again, and Chris Shaw at King's College London. And both Bob and Chris identified multiple variants, dozens of variants over time in this in FUS. Some of the variants scattered across the protein, many of them in the C terminus, were responsible for more or less typical adult onset ALS, onset in the fourth and fifth decades of life with a more or less typical progression. But there was a series of variants within the extreme C terminus in or around the PY-NLS nuclear localization signal, the extreme end of the protein that was responsible for a particularly horrible form of ALS. It's a horrible disease to begin with, but these are particularly troubling, aggressive forms of disease with early onset and very rapid progression. So for me, the first FUS case that I saw ever was more than 10 years ago now, in September of 2013. Eitan Jacoby is the boy's name. His parents like when I use his name. He was 11 years old at the time of symptom onset. Developed some arm weakness, didn't make much of it, but it progressed rapidly over the fall of 2013. It brought to us in December of 2013 suggestion of ALS, but of course, 11-year-olds don't get ALS, so he couldn't have ALS. But indeed he did. Genetic testing revealed that he had a mutation in FUS, a P525L mutation that Bob and Chris had already described. He was heterozygous for this missense mutation, which was known to be pathogenic, autosomal dominant in its mechanism. We sequenced both of his parents, both were gene negative. This was a de novo mutation in FUS. And like the other FUS kids reported before him, Eitan progressed very rapidly through the early part of 2014 and died just weeks after his 12th birthday. So his parents made the very difficult decision to donate his brain and spinal cord to autopsy. And indeed, the postmortem examination confirmed the diagnosis of juvenile onset ALS. And you can see images here. In the cerebral cortex, there was widespread neuronal cytoplasmic basophilic inclusions, these coarse aggregates. You can see them here in a silver stain of a corticospinal neuron. And in the spinal cord, there was really severe, impressive neuronal loss throughout the anterior horn. And in the few remaining neurons in the cord, we saw these FUS-positive basophilic inclusions, FUS positive with an antibody here that we generated in the lab that showed the really massive cytoplasmic aggregates in these neurons, and importantly, TDP43 negative, which distinguishes only about 3 to 5% of all ALS cases. So these aggregates, these FUS-positive aggregates, are a pathological hallmark of FUS ALS, and you'll see them again in a moment. But this was, you can imagine, to me and my team, it was a difficult, disturbing case to lose somebody so young. And we took this into the lab to begin to study the biology of FUS ALS. And we generated a series first of transgenic and then knock-in mice, which is all published now. So I'll just summarize the work by saying that we were able in a knock-in mice to show that expression of mutant FUS in vivo led to a progressive and selective motor neuron degeneration that really, in many important ways, recapitulated the phenotype that we see in our patients. We saw motor axon withdrawal. We saw, selective vulnerability of functional subtypes of motor neurons, fast twitch versus slow, alphas versus gammas. We saw evidence of both cell autonomous and non-cell autonomous mechanisms of disease in these models. We were able to show through the study of knockout mice that loss of function was very well tolerated, and that loss of function in motor neurons and more generally was insufficient sufficient to cause the neuronal degeneration that you see in FUS-dependent ALS. And through dosing experiments in the mouse, we were able to show that this was a toxic gain of function, and this was a dose-dependent phenomenon. And altogether, this led really to the suggestion of a therapeutic strategy by which lowering levels of FUS in the mouse and ultimately in the patient might, have an important effect on the phenotype. So I approached Ionis Pharmaceuticals. They had no plans for a clinical program in FUS, so they had a number of tool compounds, and they were very generous, I have to say, and shared these compounds with us, made them available to us for study in the mouse. And long story, but this initial effort led to the identification of ION 363. This was a non-allele selective gapmer, standard 2' MOE modified gapmer ASO that targeted a sequence shown here in exon 6 conserved between mouse and human. And we took this first into the mouse and then into the clinic. In the mouse, what we were able to show is that a single injection of this ASO, ICV, into newborn knock-in mice led to a very significant decrease in the levels of FUS protein, both wild type and mutant protein, as is shown here. This was shown in the cervical spinal cord, but this was true throughout the neuroaxis. A single injection of 20 micrograms of this ASO led to a 50 to 80% reduction in FUS protein in these animals. And in another model, fast-progressing model of the mice, we were able to show, more importantly, that the single injection led to very important phenotypic benefits in the mouse, both in terms of the motor neuron counts, which were completely rescued at 4 months and even at 6 months of age of the animal, when we were already seeing 15 to 20% loss overall and upwards of 50% loss in the most vulnerable of the motor pools. We also saw a complete rescue in terms of denervation of one of the early signs of the phenotype was axonal withdrawal from the neuromuscular junction. That was completely rescued at 4 and even at 6 months. And we saw also benefits in terms of inflammation, both microglial and astroglial infiltration that is, again, a harbinger of motor neuron loss in these animals. And only as FUS levels return to normal at 6 months, we begin to see the emergence of the phenotype as indicated by micro and astroglial infiltration and proliferation. So we had this preclinical evidence. We had some other toxicology data at the time, and I was very interested in moving this ASO into the clinic, and it wasn't too long before I had that opportunity. I met in 2018 J.C. Hermstad, shown here in the bottom. She had an identical twin sister, Alex Hermstad, who had the same P525L variant that Eitan had. Alex, like Eitan, developed disease very early at age 11. She was trached and vented at 12 and ultimately succumbed to her disease at age 17. JC, her sister, remained asymptomatic for an additional 13 years. And I met her because she enrolled in a program that we developed at Columbia called the ALS Families Project, which is a longitudinal study to follow gene carriers in the pre-symptomatic phase, looking for early signs of phenotypic and ultimately symptomatic onset. When I examined her first visit in this families program, her exam was already abnormal. And she had EMG abnormalities. The way the protocol is written, I couldn't tell her that at the time. We fixed that later. But it wasn't too long, a month later, essentially, before she began to develop symptoms of ALS. beginning in lower extremities with difficulty walking. By April, she was non-ambulatory. By May of 2019, unable to lift her arms, required non-invasive ventilatory support, and had severe head drop, rapid progression. Again, very similar to what we see with these P525L kids. So in early 2019, late 2018, we began our discussions with the FDA, and we began talking to them about our crazy idea with very limited preclinical data, to put this ASO into first-in-human study. They were skeptical, but responsive, and after 6 months of engagement, we got a green light to file a single patient IND, and literally a day later, we were given a may proceed order. That was on May 30th, and on June 11th, less than 2 weeks later, we began treating JC with this ASO. So JC received over a period of about 9 months, 10 months, 12 doses of this ASO. And what we saw almost immediately was that her neurofilament light levels, both CSF and in the periphery, began to come down, ultimately came down to below 50% of peak values. This is her ALS-FRS, the 48-point functional rating scale. This here is her decline pre-treatment. This is here with the first dose. And you can see that her ALS-FRS plateaued, albeit, at a low level, which may be just a floor effect of the disease, but she did better than expected, certainly better than her sister, better than Aton and others, and we thought we were seeing her to prove some evidence of clinical response. But unfortunately, at the start of COVID, JC succumbed to complications of her disease and died in May of 2020. So her family, too, made the very difficult decision to donate brain and spinal cord to our autopsy program. And what we were able to show is that the ASO was extraordinarily effective in lowering FUS levels in the brain. This is a Western blot from I think this is cortex, but the same was true of spinal cord. Greater than 90%, 95% reduction in total FUS levels in the brain, both with multiple antibodies here, including an antibody that's specific for the mutant isoform of FUS. But more importantly, what we saw was that the FUS inclusions, these cytoplasmic aggregates, and I told you are the hallmark of FUS ALS, were completely gone, nearly completely gone in this brain. I can't show you the entire axis, but it was a remarkable reduction in the levels of FUS protein. You can see this is equivalent neurons, representative, of course, from JC and these from Aton's brain and spinal cord. So the autopsy data demonstrated that we could efficiently silence FUS throughout the CNS, and this resulted in a dramatic decrease in the burden of FUS pathology in brain and spinal cord. So JC was the first patient that we treated. We renamed the ASO jacifusen in her honor, and we began to get calls from patients all over the country and later the world who were interested in participating in this investigator-initiated study. And this is an incomplete list. We've now treated over 30 patients through our expanded access program, and there are many interesting stories to tell on this list. I'll just to tell you, we've published this as a case series in Lancet last year, and encourage you to take a look at that study. But I'll tell you one story of AK Anna. that I think is particularly noteworthy. Anna is famous in her native Germany. She got a Der Spiegel piece written about her and enjoyed the fame. Anna came to us, I think it was in late 2020, right before Christmas. We treated her. Same P525L variant. She was 15 years old, maybe just after her 16th birthday. I think when she came, her mother brought her to New York from Germany, where we were unable to get the ASO in. And what we saw in the first year of treatment, as you can see here, I stole my own thunder, but that she declined very rapidly, as the other kids did. Her FRS was started at about 28, 29, and she decreased very rapidly down to about 8, had an event that led to a ventilatory crisis and requiring tracheotomy and chronic ventilation. Things were looking bad. But she continued to take the ASO. Her family was really remarkable. This is now, she had transferred her care from New York to St. Gallen in Switzerland, where they were dosing her. And Marcus Weber, who called me and said, if I didn't see it with my own eyes, I wouldn't believe it. She's getting better. And over the course of the second year of treatment, she began to recover much of the function she lost. Her FRS returned to about 23. She was able to cap her trach during the day. She again became ambulatory and living her life. She returned to online college studies. And this functional improvement, of course, was associated with and preceded by a really dramatic drop in neurofilament light levels by over 80% from peak levels in her case. So this is a video of Anna. This is not so long after she was released from the hospital. She here had already made significant improvement from her worst. You can see, I love watching her hand sort of crawl up the banister, still vent dependent throughout the day, very tentative steps up the stairs. On the left hand, right here, right, you can see Anna now about a year later, no longer vent dependent, walking up the stairs. And this was 6 months after that, walking downstairs, which was a particular challenge for her initially. And you can see her on the right now as well, walking just a little month later with greater confidence down the stairs in her hometown. So this was, extraordinary. We treated at the time initially about 6 or 7 patients, including Anna and JC. And we went back to Ionis because the FDA at one point said to us, enough, do a trial. You can't keep treating these kids through individual INDs. And the path that was laid out to me was not one that an academic physician could do. I needed a corporate partner, and that was obvious that Ionis was the people to engage with, and ultimately they decided, we had de-risked the program both in terms of safety and efficacy enough to ultimately they decided to sponsor a global phase 3 study. They called it the Fusion study to evaluate the safety and efficacy of this ASO, which was renamed ulefnersen, unfortunate name. And this is the study design. I won't go into it, but this was a 72-week placebo-controlled study followed by a 96-week open label study and a part 3, which we're in now, in many cases, open label that the center has, the Ionis has supported really generously. And this was after initial induction dose, quarterly dosing of 100 milligrams of the same ASO. And I won't go into the details of it, but this trial has now completed enrollment, and the data lock is happening in about a month, and we expect top-line data in early October, and I'm hopeful that I will be doing tours of this in the fall to report the efficacy of this drug. These are some of the primary and secondary endpoints of the study. I won't belabor them, but this was done at 20 sites, 13 countries ended up enrolling, and it really was an extraordinary effort. So I, was really impacted by this whole experience. I was proud and maybe a little surprised at what we were able to do as academic physicians. And I told you ALS is a genetically complex disorder. There are many genes that could, and many families that could potentially benefit from this same approach. But FUS was really at the limit, maybe some say below the limit of commercial viability. And there really wasn't, an opportunity, commercial opportunity for most of these patients and families. So what I knew was that, the ASOs are really, a really perfect modality, excellent modality to pursue this kind of ultra-rare approach. These are modular, sequence-defined design that allows one to achieve allele selectivity, allele specificity. We can target through the ASOs, I'll tell you in a minute, multiple pathogenic mechanisms, which we can observe in ALS. And it really is fit for ultra-rare genetic cases. This, I don't have to tell this group, knows well with these ASOs, we can achieve non-allele selective targeting of the kind that we did with tofersen and with jacifusen. We can achieve allele selective targeting either the pathogenic SNPs or a SNP that's linked to the pathogenic SNP at a locus for which the patient is heterozygous. We can use, different chemistries, but steric blocking ASOs to affect splicing, suppress mutant missense truncating exons, or suppress or encourage the inclusion of pseudo or cryptic exons. So these are all ways that we can use these ASOs in these ultra-rare forms of ALS. So the science was there, but what wasn't there, certainly wasn't there when we started with jacifusen, was the regulatory pathway to move this forward. But the FDA responded to my work and Tim Yu's work at Boston Children's and others, who pushed them to think differently about these cases. And they issued a series of guidance documents in 2021 that really govern the development of individual ASOs for life-threatening diseases like ALS. And so with this in hand, in late 21, early 22, I founded an initiative called Silence ALS to discover and develop individualized investigational ASO therapeutics for ALS patients with ultra-rare pathogenic mutations. And this was a partnership between us and the NLORM Foundation. NLORM is an organization that was founded by Stanley Crook, founder and longtime CEO of Ionis Pharmaceuticals, who did this in his retirement. And the idea really was to create an infrastructure that builds on NLORM's nonprofit precision medicine platform. And the idea was to bring together academics and nonprofit stakeholders and use our disease-specific knowledge in ALS combined with NLORM's resources to treat these underserved patients who have these rare genetic mutations. And the idea was that we would use our resources to identify symptomatic carriers of these ALS mutations, for whom really commercial program was possible, but also, as I might talk to you about later, pre-symptomatic individuals who, might benefit from early intervention even before the appearance of symptoms. So our efforts have focused on genetic mutations that cause ALS through dominant mechanisms so far, amenable to ASO therapies. That includes toxic gain of function, dominant negatives, aberrant splicing events. This has been our focus. Here are some of the genes we're interested in. I'll tell you as much as I have time for. Major focus of our efforts is TAR-DBP. We have an active program in CHEHD10, which I'll tell you most about. Active late-stage programs in the Nexin A11, and ongoing programs in a variety of other genes. This effort, I should say, has been largely in recent years funded by the URGENT network, the ultra-rare gene-based therapy network of NINDS, and we're very, very grateful for that support. But in recent months here have begun to think about loss of function haploinsufficiency states using ASOs to potentially upregulate genes whose expression is deficient in ALS, like TBK1 as an example, which is our major effort. So this is our workflow, which I'll take you through quickly. Again, as I said, begins with the patient. We do very deep phenotyping, genotyping of our patients. We confirm the pathogenic variant. We perform long-read sequencing for haplotype analysis and phasing of the pathogenic variant. And then we build in vitro models, fibroblasts, and when required, IPS lines from our patients. And then we present the case to NLORM for vet and accepting. And if accepted, we begin the ASO design and discovery program that begins with ASO design, in silico studies, in vitro and then in vivo tolerability studies. And then it ultimately leads to lead selection of an ASO that we bring into a 13-week GLP toxicology study that is mandated by the FDA, even in this limited approval pathway. We typically don't have to meet with the FDA any longer, but we write and submit INDs, in most cases individual INDs, but we've moved this now so that the FDA is accepting multi-patient INDs, which is a real, again, breakthrough, in a regulatory breakthrough, I think, in this area. And then with approval, we begin first-in-human studies. So this is just a quick summary of the patients that we've enrolled in the Silence program in recent years, 200 patients in total. The majority of these are symptomatic for ALS, some with ALS FTD. I can maybe tell you, are beginning a program on frontotemporal dementia, given that these same variants cause both diseases. We also are following a significant number of asymptomatic carriers, some of which have converted in the course of this study, and they represent a variety of genes, as shown here. These are the genes in which we're interested in and focused on. The TAR-DBP represents the majority of our cases, but about 10% each of our cases involve CHEHD10 variants, and Nexin A11, profilin is growing. And as in many of these cases, it's a build it and they will come kind of a situation where you tell people that you have a therapeutic, and then all of a sudden this nano rare becomes ultra rare and doesn't seem that rare at all in some instances. So most of our participants are from the United States, but there's been an extraordinary expression of interest from the international community, mostly from Europe, but from the Middle East and elsewhere. And as we grow the program, we're beginning to explore our efforts, as I'll tell you, to push these ASOs into the world. So I'm going to tell you about our most mature program as just maybe an example of what we hope to do repeatedly. This is a focus on a gene called CHEHD10. This is a gene in which ALS FTD-associated variants were first identified in 2014. And CHH10 is associated with the multi-system autosomal dominant disorders, including ALS and FTD, but also myopathy, Parkinson's, CMT. But there are two mutations, R15L and S59L, which are particularly associated with ALS. And so as we've done with other gene targets, we've amassed a group of scientists and clinicians interested in the biology of this, which fuels our therapeutic design and discovery. And we've already published a number of papers on D10, including this one published last year in Brain, effort led by Derek Narendra and Justin Kwan at NIH, characterizing a German-American family with a CHH10 R15L mutation. This again is published. It's actually off medRxiv now. It's in Brain. But in this paper, what we've done is characterize this large 7, now 8 generation pedigree with at least 68 affected individuals. We demonstrate very high, upwards of 80%, but still incomplete penetrance of this variant. And what's extraordinary is, to at least the neurologists and the audience, is that this is a highly stereotyped phenotype, and the most stereotyped of I think all the rare genetic cases of ALS that I've seen. These patients invariably present with flail arm syndrome, brachial amyotrophic diplegia, or amyotrophic diplegia, suggesting greater involvement, from the phenotype of the cervical spinal cord. And so, because we're following these families, we've fortunately and unfortunately gotten autopsy materials from these cases. And what we were able to show, this is Vlad Korobeinikov in my group, demonstrated subtotal loss of motor neurons throughout the spinal cord, and relative sparing of lumbar segments, as one would have predicted from the clinical phenotype. Prominent CHH10 protein aggregation, you can see staining here, filling the cytoplasm, but then you can see these sort of globular aggregates as well. And importantly, again, absence of TDP43 pathology in this case, one of the rare non-TDP43 cases. We modeled these in fibroblasts and iPSCs. We created knock-in models. And what we were able to show, again, as we did with FUS, with this loss of function, was highly tolerated. And this really seemed to be another toxic gain of function, which suggested that we could approach this using a non-allele selective approach, as we had previously. And with our friends at Lorem, developed NL-CHH001, a non-allele selective ASO. Again, same chemistry as modified MOE, 2' modified gapmer. And this was an ASO that we brought through tolerability and toxicology studies, and we received our first, about a year and a half ago, IND to proceed with a single member of this family, shown here. And we pretty quickly extended to treat 3 of his other affected siblings. And then we began branching out across the pedigree to treat, increasing numbers of family members. Now I think up to 16 family members have been treated with this ASO. And here's a list of these patients treated at a variety of centers now across what we're calling the Silence ALS network. But it's, as I told you, a German-American family. And we traced this variant back to Germany, to an area outside of Bonn, where the family was originally from, and identified several patients with this same variant. And together with my friend and colleague, Patrick Veit, treated our first patient last week with this same ASO. And there are several other patients in Germany with the same variant, and some that have, strayed across the border into the Netherlands and into Northern Europe, that we hope to treat with this ASO. So to what effect? So we treated these patients with an ascending dose. Each patient received 3 doses of 50 milligrams, followed by 3 doses of 75 and up to a final target dose of 100 milligrams. Between each dose, of course, between each dose escalation, we were reporting back to the FDA to get an okay to proceed. Now with over, I think now closer to 70 individual doses, we've seen no drug-related AEs. One death, sadly, one of our patients developed an RSV pneumonia. We think this was unrelated to the drug, but the ASO is proven remarkably safe. This is our combined NFL data. You can see that we have treatment out to almost closer to 2 years now, but 18 months at least. And this is the NFL data from these cases. You can see overall a downward trend in NFL levels quantified here. Average drop across all patients, including those that have been only recently treated, a 45 to 50% drop in NFL levels in the CSF. And the patients treated the longest, we've seen a 61% drop after 572 days of treatment. So we're seeing consistent, even at the low dose of 50 milligrams, we're seeing, I would say dramatic and significant decreases in neurofilament. With the one autopsy case that we saw, we were able to show target engagement, CHGH10 levels, which are elevated in D10 patients over controls, was dropped down to near normal levels, we believe, in this one case. And of course, we have to demonstrate this further. So, what are we seeing in terms of clinical benefit? Not clear yet. There are some who want to claim this as efficacy. I'm withholding my judgment, both in terms of FRS, which you can maybe squint your eyes and begin to see some inflection at treatment, and in forced vital capacity. There's some suggestion from our various measures, but this is going to take longer given the relatively slow rate of progression of CHGH10 ALS. Going to take some time, I think, to demonstrate efficacy, but we got a grant early this year from the DoD to launch a 2-year phase 1/2A pilot clinical trial of this ASO in about a dozen patients. Hopefully we can add some numbers. And this will occur at Northwestern, Columbia, and the University of Minnesota. So this will take some time. The NFL data alone, I think, is pretty significant. And in the wake of the FDA's recent issuance of the guidance on plausible mechanisms pathway, there's some would argue that we have enough data already in this population based on NFL alone to apply for accelerated approval through this guidance. And it's something that we're considering, and that may be a pathway forward for therapeutics in the ultra-rare space as we go forward. All right, so I think we're okay for time, yeah? So I just wanted to end by talking about a major effort of Silence ALS focused on TAR-DBP, which is the gene that encodes TDP43. Mutations in TAR-DBP associated with ALS and frontotemporal dementia are scattered across the protein, but mostly in the C-terminus of the protein. There are dozens of these, as was the case of in FUS and SOD1. It's a common story. TDP43 has been for now a long time recognized as a major player in the pathogenesis of the disease. Certainly, it is a hallmark, a pathological hallmark of ALS and many forms of frontotemporal dementia. And we see, as you can see here, these cytoplasmic inclusions, these large globular inclusions, skein-like inclusions, various morphologies that you see in different cell types throughout the brain and spinal cord. There are those who believe, I believe, that these are pathogenic, and that their presence is toxic to neurons. And the idea in approaching this was that by, as we did with FOS and tofersen and CHH10, was to lower levels of TDP-43, might allow us to achieve the kinds of clearance of this pathology in TDP-43 cases, which again represent something like 97% of all cases, including sporadics, at least in the gene carriers. Lowering FOS levels, the idea was would help to achieve some rebalance and elimination of this pathology. But there, and this is just our cohorts. Most of our patients, 2/3 of our TAR-DBP patients are symptomatic with ALS, few with FTD as well. A third asymptomatic, variety of variants that we deal with. So again, the pathological hallmark of the disease was this these abnormal cytoplasmic inclusions. The problem is that associated with this is nuclear exclusion of TDP-43 and functional loss, nuclear functional loss of TDP-43. And there is a lot of interest in this idea that TDP-43 does a lot of things. It has many roles in RNA metabolism, one of which is as a splicing regulator and controlling the inclusion of cryptic exons. And when TDP-43 is excluded from the nucleus and there's a release of its regulatory role, and so you end up getting cryptic exon inclusion that can either lead to nonsense mediated decay and elimination of proteins, or the inclusion of cryptic exons in frame that result in neopeptides that might disrupt the function or create novel functions that are toxic to neurons. So there's a real concern that TDP-43 loss of function could be contributing, if not primarily, secondarily, to neurodegeneration in ALS FTD. And so, it made it difficult for us to think about targeting this gene in a non-allele selective way as we had with CHH10 plus SOD1. So the approach that we've been taking largely, but not exclusively, in TAR-DBP is to develop allele selective ASOs. And this is just an example of the first case. This was a 40-year-old French-Moroccan woman with spinal onset ALS, October 2020. And we demonstrated that she had a rare de novo, both parents being negative, pathogenic mutation in TAR-DBP. And over 11 months, we haven't beaten that time since, but we developed an ASO called NL-TARD-001. This was an allele selective ASO targeting a sequence that was unique to her mutated allele. It had 55x selectivity of the mutated allele over the wild type non-mutated allele. And we got IND approval to treat her and began treating her. She didn't get very far, unfortunately, unrelated to the ASO. She had an event that made it impossible for her to return from France to the United States, and we ended the treatment quickly, but we soon treated a second patient with this same ASO. More complicated case, we can talk about it, but we did treat her over the course of the better part of a year and saw a 20 to 25% decrement in neurofilament light in her case, which we found very encouraging. Her FRS, the second patient, was very low, and our objective and the objective of the family was largely to get safety data that we could move forward as other members of the family might be affected. And it was a useful exercise, at least to know that treating people with a allele selective ASO, TAR-DBP ASO, didn't result in increase in NFL, and there was those who warned me not to do this because there was danger that we could induce a deficiency state or exacerbate a deficiency state that would make the phenotype worse. So there was a lot of discussion about risk benefits in this context, which I'm sure we can talk about further. So when I began this TARDI-BP program, I really thought that I was going to have to develop individualized ASOs for every patient to get an allele selective drug. It thankfully turns out not to be the case. You know, at $1.5 million a pop, I really am glad that isn't the case. And we're finding as we do sequencing on more and more gene carriers that we are able happily to use these same ASOs again and again in patients, unrelated patients with different variants and different haplotypes, but we're able to use these in a non-allele selective way in these different patients. As an example, TARD-005, which is an ASO that we hope to treat a patient with here in Rochester in the coming weeks. We're treating, well, 3, but there's actually 5 patients now that we have identified that can benefit from this ASO. 06, there's 2 here, but we have 8, I think, patients that can benefit from this ASO. We've identified in a allele selective manner, you know, same as true of other ASOs that we're developing. So that's very good. So this is a list of the ASOs that we have so far in the clinic. The D10 ASO, we have multiple 01, 02, 05, 06, hopefully soon 07 or its replacement, and others in late stage development. These are ASOs that are already in clinic for which we have INDs and can apply more broadly to the TARD-BP community. And we have late stage programs in annexin A11, which is non-allele selective, and several other genes coming along, gene targets coming along that we hope to be able to scale. So I'll end by saying that you know, we have a lot of ambitions for this program for Silence ALS. Our hope is that we can scale this initiative, involve more patients, additional ultra-rare ALS gene mutations, both with gain and loss of function mechanisms. We are building our network of ALS clinicians who are interested in participating in this both nationally and internationally. As we do this, we're gaining efficiencies and both in cost, which is not unimportant, and in time, and the ability to apply ASOs, you know, to patients and not have to stand up an individualized program for every patient is creating huge efficiencies now in a program, and I'm hoping that that will just build in time. We now have a small but growing library of Silence ALS ASOs that I said we're going to reuse for patients with shared haplotypes and targets, and we're really beginning to focus on pre-symptomatic carriers. We're sequencing them. We're doing the haplotype analysis and phasing that's needed, and we're trying to come up with a plan where we have, in anticipation of phenotypic conversion, an ASO that we know if this patient does develop signs or symptoms of disease, we can, you know, quickly intervene with an ASO, ideally before the symptoms develop, this is something I think that we're going to have a lot of opportunity to do as we move forward. And I think this is, you know, the greatest hope that these kinds of therapeutics have. They're, you know, I think I always say the art is in the prevention, and that is very much true in ALS, sporadic and genetic forms of ALS. And then, you know, as I've spoken to many of you about my talks over the last couple of days, we're looking to extend our efforts beyond the rare genetic forms of ALS into frontotemporal dementia. That's a relatively easy one. We have a protocol now that we've developed with our FTD colleagues, and you know, as we follow these pre-symptomatic carriers, a good percentage of them develop ALS, but some develop frontotemporal dementia. And there's a lot of neuromuscular people here. You know that the last thing a neuromuscular doctor wants to do is a cognitive behavioral assessment. So there was no way I was going to do that study. But I've involved my colleagues in it. They seem to be very interested, and we have now a plan to move these ASOs into the FTD realm. But you know, as I've said, that the genetics. People will often say to me, you know, this is all great, but you're talking about 10, 15% of patients. What about the 85, 90% of patients out there that don't have a pathogenic variant? What about us? And my, answer to that is that, there is a shared biology of disease. The genetics are telling us something about that shared biology. And I believe that many of the targets that we're going after in these familial forms of ALS are relevant. TARDBP is an obvious example, relevant to the pathogenesis of sporadic ALS. And I think, we have a regulatory pathway to do what we're doing in the rare genetic forms of the disease, but it's only a matter of time, I think, before we're able to apply this to larger populations after we've demonstrated proof of principle, safety first and proof of principle. And that ultimately is my objective to move these forward into the non-genetic forms of the disease. And we've already had through, some of our preclinical work, evidence of functional interactions between these ALS gene encoded proteins that suggest, that we're only beginning to understand the nature of this shared biology. And I think there'll be a lot of opportunity to move these drugs, forward on a larger scale. So this is our, Pete has this email, so I don't have to share it too much, but this is Ellie Harrington, who is responsible for so much of the Silence ALS program and a point of contact for anybody who has a patient with a variant that might benefit from one of our drugs. And as I said, we're working with Pete, and Emma's been very helpful in this. We talked about it today to help and Bob, thank you, to get Rochester up as a site within the Silence ALS network and a referral site to treat these patients. And we're hoping to build a network nationwide, but also internationally, of sites to develop these and move these ASOs forward. Lots of people to thank. In my lab at the ALS Center, Matt Harms has been my partner in so much of this. Special thanks to him. I have colleagues at Ionis Pharmaceuticals that have been extraordinarily generous throughout this now 7-year FUS ASO experience. I mean, this is a group, we give them a lot of shade, but I think that they are really focused on the patient and the needs of the patient, and have been just wonderful partners through it. Ionis has now licensed the drug to Otsuka Pharmaceuticals, which has also been wonderful partners, but they don't get a listing yet. And Lorem, Stan Crook and his team are, extraordinary, and, hoping to build on that relationship. Our growing network of sites, the blue represents our international partners, multiple funding programs. This has been a multi-million-dollar effort in requiring a lot of federal, private, foundational, and government support. So thanks very much for listening. I'm really happy to take questions. And thank you again.
Well, thank you, Neil. That's really transformational work. Congratulations. So just, I see Mox online. I don't know whether you have any questions. If not, I know I have a couple, and I'm sure others do.
I'm going to let you ask the questions, David. I'm just overwhelmed by the opportunities, and just hope that there's ways of networking with Dr. Shneider's group and any other group that's pursuing his discoveries. And just my thanks to him and to all those who've made the lecture possible.
Well, thank you, Mox. So Neil, help me understand. So the neurofilament light chain seems to start dropping pretty early in your patients, yet your patients continue to progress. Mm-hmm. And we sort of maybe saw a similar story with SOD1 and tofersen, although people look closely at those graphs, maybe things were continuing to plateau and then the clinical improvement came in the months and years to follow. What is it why are patients continuing to clinically progress? What have you learned from Anna? Do you understand that sort of link between neurofilament dropping and then what we may hope for clinically?
Yeah, it's a great question. Bottom line, I don't know the answer, but neurofilament is a measure of what exactly, I don't know, neuronal injury. But what we understand surely from about ALS is that there are neurons that are gone and aren't coming back. There are neurons that are vulnerable and dysfunctional, and then those that are vulnerable and not yet affected, and those are the ones that are trying to respond as best they can. Those that are at least vulnerable to sprout, and they're the ones responsible for the reorganization of motor units and of physiology that we see in ALS patients. And so, I wish, and I was talking to Rick about this earlier today, and I really wish that we had done motor unit studies on Anna as she was getting better and seeing what was going on, and that's planned for future studies. But what I would say, little hand waving, is that we're taking our foot, let's say, off the end of the first gas, right? Or that's what you want. Like you're lowering levels pretty quickly, I would say. But the aggregates are still there. They're still doing damage. We know from the first case that there are homeostatic mechanisms in place that can clear those aggregates. I mean, I presume that JC had aggregates. We didn't see them at her autopsy, but it's because they were cleared. What the ASO does is, and we're hoping to do, across the boards here, is to aggregate formation is input and output, right? That you have aggregate formation and you have aggregate clearance, and there's an imbalance of that, obviously, in the case which results in the accumulation of these toxic aggregates that kills neurons. You stop the production of it, you give autophagy and other proteostatic mechanisms an opportunity to clear those aggregates and to begin to restore function. Now, if you have any ASO, if you have any motor neurons left after all that's done, you might actually see some kind of real functional, and long-term benefit. But in the pools where the neurons are gone, we ain't gonna help them.
Yeah.
Those pools are gone and not coming back. But in the pools that are not yet affected, that's why it's so important to get in early. Is we can shut this off and alter the course, before you reach that point of no return. That's my thinking. I think that we can figure out ways, clinical and other ways, to provide evidence to this idea. But we're already in mouse models, achieving some understanding of this as we do single cell and other multiproteomic analyses of neurons as longitudinally through the course of disease. You see things changing, right? And you need to intervene earlier for, and the nervous system, it takes 6 months, at least a year plus, right? For the neurons to die. You don't shut it off and expect immediate, recovery. There's a process, and then we're learning now for the first time, utophin, jacifusen, and hopefully others, what success looks like in ALS and what we can expect in terms of clinical benefit. And frankly, it's because of our work, Tofersen and our work, that people are thinking differently about ALS trials. I mean, the idea that you can do an ALS trial in 24 weeks, expect to see a functional, benefit, is this being broadcast? Is laughing, right? In the face of this data, right? NFL levels might drop. They drop very, quickly. That's a good sign, right? Maybe a harbinger of clinical recovery. But you can't expect the nervous system to recover in 24 weeks in ALS. It just doesn't, not possible. It just, defies the biology. Anyway.
Alexis?
Thank you for a wonderful and inspiring talk. And I think it speaks to something very deep in many of us who chose academic neurology and neuroscience. Very impressive to me is also the infrastructure around a program like this. And I'm just wondering, I remember with JC, it was quite a protracted time until she had the ISO administered, right? Have you seen those timelines shorten up, or are there any lessons in kind of getting these things off the ground quicker?
I mean, there are holdups at multiple stages. And you know, in our program, we have Columbia, we have Eleanor, we have FDA, we have IRBs. You know, so I would think it could be quicker. When I had control, but when I was using the Plus ASO and expanded access program, I could get somebody treated because I had full control I submitted the IND, I submitted the IRB, I controlled the patient. I could get somebody treated within a month of submitting the IND. It should be that quick, but that's why we need. But that's true. The FDA is a little slow these days, but that's why we need approvals, right? So, and even in CHH10, some of our patients, a variety of reasons, take 6 months, a year to get treated with our ASO. And so, what I want to do is take these ASOs, get the approvals, put them into, you know, get some new co or some entity, you know, to support this, and then just treat. You know, somebody's diagnosed with ALS, probably have the pathogenic variant, they get treated like you do with the SOD1 cases. How long does it take to get somebody approved for SOD1? Not 6 weeks.
Depends on the insurance.
Yeah.
Thank you.
Anyone else?
I have a question. Thank you. Wonderful. I have a lot of questions, but I'll save those for later. But here's one. I'm piggybacking on kind of this concept of earlier is better and neurofilament, right? And so just thinking about how do we do this? When's the right time? How do I identify that right time? Because people can certainly have pathologic mutations for a long time before they become symptomatic. And, you know, I wonder if maybe you've been able to get some information on this just because of the families you've had. You know, I know in the field and some approaches have largely relied on NF levels. You know, do you do percent increase? Do you do a threshold? And then you say, this is when you treat. Makes sense, but I wonder if we could do better. And I wonder if in your case you have families. JC's an obvious example. Her sister Alex had it 7, 8 years before. I wonder if, you know, it could be more than just purely changes in NF, but it could also be perhaps a pretest probability or a risk where you have this genetic variant that then lowers your potential threshold for a change in NF, or perhaps another model. Have you explored this with some of your families and thinking about, you know, how do we optimize timing of initiation in a pre-symptomatic prodromal patient?
It's a good idea. I mean, you know, I think it's hard to model. JC and Alex were identical. Yet their onset was 7 years apart. 13. 11 versus. Go figure, right? I mean, model that. But there are, you know, examples. That list I told you, there were stories. One of the cases was a man who from a P521 L family, our 521 L family, who mother died, therapeutic divorce, onset in late 30s, early 40s, dead by 2 years old. We met this man. He had physiological changes, sharp waves scattered, not diagnostic. Like maybe elevation in neurofilament level. Wrote to the FDA, and I said I want to treat this from this family. And I was not expecting they know, but they and we treated him. That was 4+ years ago. Both of his sisters died. They were relatively late in their course, and he is symptom-free. His EMG normalized after a year. His NfL's normalized. He's lived 4 or 5 years, 4+ years on treatment without developing symptoms. And you can tell me, well, that doesn't fluid make with him anyway. Well, that you take this. So and he's profiled on CBS News. He's quite a character. But living his life as a carrier on drug and has modest levels of so I think that with vigilance and better markers, neurological, molecular fluid markers, we could come up with a strategy. But first we have to demonstrate, and there are many people, I should say, in the prosecution trial who were like this guy, who were being followed in programs, who triggered the inclusion criteria of the trial. Very little. A lot of people in the trial were pre-symptomatic. And there are several of those. That's going to complicate the analysis. Worried about this, but because they have normal FRS, they have normal FBC, they're following them, nothing happens, then it becomes open natural history, right? Like you're not seeing any decrement in their FRS, change. You're not seeing any clinical improvement. You're seeing that they're stable, which maybe we can, find statisticians can figure out how to use it. Anyway, I mean, I think there's lots of evidence that these work now in symptomatic individuals, these drugs, and then how we apply them, the best way to apply them in other forms, it would be different, probably. Benign, different, probably BP, different.
Wonderful. Bob?
Just tremendous, so inspiring again. Just how many, there's about 5,000 new patients per year with ALS in the United States. And in a couple of years, what's going to be the cost of whole genome sequencing? You think? Yeah, a couple of years, how much is going to go down? $50. $50. I mean, what, because all when I'm hearing these talks like this, I'm just like, it's just like lowering the threshold for like, hey, let's just have to cap the population. Why don't we start? How do we expand doing whole genomes on all of these patients? And how can the community start to work together to do that? We're so focused on trials, but what's the scaffolding to think about? Just then with all of us coming up and just, I mean, I'm a little naive in this space, but I love it. But I'm just thinking like, why aren't we doing more whole genomes on everyone and creating an infrastructure for polygenetic mechanisms and disease, rare ultra disease? But it just seems like there's a space there that we got to be starting thinking to get into. And I mean, maybe that's really naive. It's like, how can we keep it? It seems like, wow, why wouldn't we be doing that?
No, I mean, there's this is a huge topic, right? People are having a lot of discussions, ethical and otherwise, about this. But I mean, I would, at the very least, like in the FUS case, would like to see more testing in young populations. Like in FUS, for many of the truncating and extreme C-terminal pediatric juvenile variants, there is a prodrome of tremor, neurodevelopmental delay, and that I think is associated with partial loss of function, which is happening in addition to the gain of function, which is the cause of the ALS. But so these young people who have individual learning plans and issues should be sequenced for FUS mutations, and it's not subtle. I mean, all of these P525L kids have some degree of neurodevelopmental delay, and there have been a couple of cases where we've identified kids pre-symptomatically. Well, they have the neurodevelopmental issues, but they don't have ALS. So, I think we start small, right? You begin, I'm not sure everybody's ready to have a whole genome sequence at birth and have all of the, deal with all the implications of that knowledge. Some people just don't want to know. So there's, yeah.
Interesting.
Sure. Yeah, anyone on the line with any questions? I want to see the chat here.
You're not in there. Yeah, you're not in the zone.
Yeah. Yes. Oh, so there is one online question and comment thanking you for a wonderful presentation. But they were asking, I was curious, as more treatments using ASOs are coming up, how difficult will it be to determine dosing duration for the different ASO treatments? Is this a bottleneck to starting trials?
Dosing duration.
I'm wondering whether it's intervals.
Intervals,
doses,
yeah. Yeah, well, that's, true. It is true that we don't know exactly how to use these drugs. And we're going to have to learn over time. I believe with FUS as an example, that, you have 95%, plus decrement in NFL, in FUS levels. Do you need to keep that up, after at some point? Maybe you can allow FUS levels to return to some reasonable level, 20%, 30%, that would, still be below some threshold for, the saturation concentration of this protein to cause aggregates, but, not so low that you're creating deficiencies, which could be a real concern for patients long-term. And that's part of the argument for why don't we just treat all carriers? Well, what do I know about treating somebody for 20 years with an ASO inducing this kind of FUS deficiency? Nothing, right? So, but maybe there's a different way. Maybe if you drop your NFL level, if your FUS levels or SOD1 levels or whatever target of choice by 50%, maybe that's enough, to really reduce risk, to make a disease with an onset in the 4th and 5th decade into something that's, in the 8th and 9th decade of life, and that'd be pretty good. So I think we have a lot to learn about how to use these ASOs, and, tofersen is not the end, it's the beginning. It's just like with nusinersen. I mean, there's going to be a series of drugs coming out, I'm sure, targeting SOD1, different chemistries, different modalities that are dosed at longer intervals. Tofersen's monthly, it's really onerous. But twice a year, once a year dosing of these drugs is going to be, big change. We achieve proof of principle, and then we do the work to figure out, I think, how to use these drugs better.
Well, thank you, Neil. This was a wonderful, wonderful presentation, and certainly learned a lot, and we look forward to continued exciting developments in the plethora of therapies in the years to come.
Thanks, David.